Hello student, today I am taking next lecture on different method of asexual reproduction. In last lecture yesterday I taught you the regeneration, spore formation and budding. After that the next method of asexual reproduction I am explaining today. It is vegetative reproduction. Vegetative reproduction is a method of reproduction in which we produce new plant by the use of any vegetative part of the plant like stem, roots and leaves. I am writing the definition. It is a method of asexual reproduction in which we use any vegetative part of the plant for production of new plant. The vegetative part can be leaves, stem and roots. So when we use any vegetative part of the plant like stem, roots and leaves and we form a new plants, this method of asexual production, it is called vegetative propagation. You know, plants contain two parts. The flower is the reproductive part of the plant and except flower all the part of the plant are called vegetative part. So when we use any vegetative part for the production of new plants, this method of reproduction is called vegetative propagation. Vegetative propagation is divided into two categories. Number one, natural vegetative propagation. In this method we use different part of the plant. There is no any involvement, there is no any use of human methods. Why the root, why the using of leaves and stem, the plants produce new offspring naturally. There is no any involvement of human beings, so there is no any role of human beings. They naturally produces new offspring and new plants. This process is called natural vegetative propagation. Second, artificial vegetative propagation. In this method the human beings involved and they produces new plant by the utilization of different part of the plant. An artificial method of vegetative propagation is also known as man-made method of vegetative propagation. There are following part of artificial vegetative propagation. First, cutting. Second, layering. Third, grafting. Layering is also known as Guti. In cutting method, we cut any twig of a stem of any plant and we place this twig of the plant in another place inside of the soil. Then this twig form a new plant. This is called cutting. In layering method, we give the cut in any aerial branch of the plant. Then after we place this cutting part of the twig inside of the soil, and after some time we watch that there is development of roots. Then after we separate this cutting part from the parental plant, then after this cutting part form a new plant. This is called layering. And air layering, we do same things in the aerial branch, in the ear part. In grafting method, we take two plants, and we give the cut. We take two twig from the two different plants, and then plants can be same also. Then we give the complementary cut in both the twig, and we fix both the twig and wrapped by a thread, and we place the complex twig inside of the soil. Then one twig of the plant form root and the twig of the plant form shoot. So which twig form the roots of the plant, this is called stalk, and another form the stem, this is called scion. So this is artificial vegetative propagation. Next method of asexual production, micro propagation. Like I think 6, micro propagation. It is also known as tissue culture. Micropropagation in tissue culture is also a technique of formation of new plant. In this method, we take very small part like some cells and tissue from the plants, and we place these cells and tissue inside of a culture medium. Then inside of culture medium, then these cells start the cell division and form a bunch of the cell. This bunch of the cell is called callus. Then this callus we shift inside of the soil, and it start the differentiation and form new plant. So in this method we have watched we are using very small part like tissue and cells for the propagation of new plant. Due to this reason it is called micro propagation and tissue culture. It is used by the scientists for the production of new plants, those species which are in endangered range. So we save protoplasm and some cells of these plants. Then after we can utilize these cells for the production of new plants in future. So this is very good technique nowadays for the production of new plant. Thank you.